Okay, so here we have 2N diploid cell where N represents 2 because we have 2 pairs of chromosomes, one from the father and one from the mother. And this cell is about to go through meiosis, which is the division of one somatic cell to create 4 1N haploid cells that are genetically different from one another.
Okay, the first phase of this process is interphase. Now, interphase is when the DNA in the cell replicates itself. So now we have a 2N cell, but it has the copies of its DNA, so it could be referred to as a 4N cell. So next, the cell goes through synapsis, which is when the chromosomes switch segments of DNA, so they go on top of each other. So they switch segments. So now the chromosomes have finished going through synapsis, so you can see that they've switched their segments of DNA, so every chromosome is different.
Now the cell is going to go through prophase, which is when the chromosomes become visible, the nucleus disappears, and the spindle fibers appear.
So the next step is metaphase, which is when the sets of chromosomes line up down the center of the cell. So the long pairs of chromosomes line up across from the long, and the short line up across from the short. Also, the spindle fibers attach to the centromeres of each set of chromosomes to prepare them for anaphase.
Now, anaphase is when the spindle fibers pull apart the chromosomes and they disappear.
Next is telophase, which is when new nuclei are formed, and then cytokinesis happens, which is the division of the cytoplasm. So now we have 2 cells which are not genetically similar. They are genetically different because of the synapsis, which is one difference between meiosis and mitosis.
Each cell will divide again, so we again go through synapsis. Where are they? Now each cell just went through synapsis. Now each chromosome is even more different. And now each cell is going to go through prophase.
Phase, which is the lining up of the chromosomes in the center of the cell, and the spindle fibers appear at opposite poles of the cell to prepare themselves for metaphase.
Now each cell is going to go through metaphase, which is when the chromosomes line up in the center of the cell, and the spindle fibers attach themselves to each centromere.
Next is anaphase, which is when the chromosomes are tugged apart to opposite poles of the cell by the spindle fibers. So they're pulled apart and the spindle fibers disappear.
Next is telophase, which is when each set of chromosomes are at opposite ends of the cell and a new nucleus forms on each set of chromosomes.
The next step is cytokinesis. So this is when the cytoplasm pinches apart and creates 4 genetically different haploid 1N gamete cells. So now you can see that we have 4 1N genetically different haploid cells, each with 2 chromosomes, and that is how gametes are formed. We started with a 2N diploid cell, which N represented 2, and now we have 4 genetically different haploid cells, and that is meiosis.